Oh, no, I don't know why I said that. I think it's because the other girl,
All right, how's everybody doing? Good. Wrapping up the fourth week of the semester. Hopefully, some of the early semester weeks, some of the anxiety is starting to wind down, or I guess maybe things are getting kind of wound up depending on when exams are early this semester. So I'm going to pick up where we left off last time and go after wrapping up some of the ways that we measure virulence and what techniques that we use in the lab. But before we do, I wanted to start off with a quick question. That is the following. So if, when we're thinking about ways to quantify virulence, last time there were a number of different assays, and one of them was the competition assay. And this is a currently favored method of measuring virulence in vivo. So like looking at how virulent a particular pathogen is in an animal host, competition assay is far and away one of the best assays that we have. And why is that? And so you might think that A, estimating the time to death of infection in animals is more accurate using this method. Estimating within host fitness of pathogens is more accurate using the competition assay. Estimating the lethal dose of a pathogen is more accurate using this assay. Or that estimating growth within the host is more accurate using competition assay. Or estimating localization within the host is more accurate with this method. So I want you to think about why this particular method is so good. And go ahead and fire in your answers when you guys are ready. So windows open, discuss with each other, and then answer with me.
Yes, so now issues of the one and are compared against each other. Describe the fitness of the substance. Yes, and the competitive index and mutant are more the competitive index is higher than one, but the control strain is more it's like we have I thought you really like you can't calculate it. Yeah, they just aren't. And then she your chances of I mean, I'm like, this is Wilson protecting it. Like I do. The only thing I can't get is I don't have
Go ahead and get back to the business at hand. And that is wrapping up our discussion on measuring virulence. So virulence, we basically give a whole bunch of approaches for studying virulence in whole animals. But sometimes working with whole animals is not really letting you get at exactly what you want to get at. Things like adherence to certain host tissues, for example, and exactly what are the molecular players in this host pathogen interface, this interaction between host and pathogen. A lot of the cellular mechanisms that we're going to talk about, things that, like say salmonella is tricking host epithelial cells into doing, we've learned most all of this by working in tissue culture. What tissue culture is essentially mammalian cells of different varieties, essentially different differentiated cell types, epithelial cells being one, that we're growing these in culture. So we're growing them outside of a host. And then there's also organ culture. So this can be whole or partial organs. So if you can't have a particular cell line that is derived from, say, epithelial cells or kidney cells or liver cells, if you can't work with that, oftentimes you can work with harvested organs from an animal. and essentially look at what a pathogen is doing to a particular organ tissue. So like looking at E. coli interactions with bladder epithelial cells and things you can do with harvested bladders. So what do you get from doing this that you don't get from studying things in whole animals? Well, you get things like this, right? That you can really look at where is this pathogen interacting with either an artificially kind of grown layer of epithelial cells, or if I've harvested epithelial tissue from a mouse and I were to expose this pathogen to this tissue, what are they adhering to? How are they doing this? How are they sticking around to cause disease, right? So that's not something we get looking at whole animals. Invasion of host cells. So this is going to be a major important process we're going to talk about in the next kind of module, we might say, in the course. And then finally, toxicity. Quantifying how nasty a pathogen is in terms of its toxicity to host cells. This is something that you can't really do in the animal. Because the animal, what you're looking at is the manifestation of this disease, right? You're looking at essentially an integrated phenotype. So this pathogen is causing all kinds of problems. A lot of problems we'll talk about, but really, you can't pick apart pieces. You can't be very reductionist looking at what the host is doing or the pathogen, looking at the whole animal, right? So that's one of the things that tissue culture really gives us the power to do. Not that it doesn't have its limitations, which it really does. And I'm going to enumerate some of what these are. And in particular, this first statement is that immortalized cell lines are genetically and sometimes morphologically altered. So if you're talking about HeLa cells, if you're talking about HEK cells, if you're talking about Keiko cells, whatever human cell line that is really commonly worked with, that a lot of people use to study pathogens, what those are is they are immortalized cell lines. Okay, so they have been essentially fused with a cancerous cell line, and essentially they are essentially cancerous epithelial cells, right? So they grow uncontrollably. So they have a broken set of cell cycle regulators, and so they are just proliferating indiscriminately. So the benefit with that is that we can grow them in the lab, and we can grow a ton of these cells. And they look something like the tissues they were derived from. Now the problem is when we make things, when we immortalize cell lines, the problem with that is that there are a lot of other unintended consequences that come along with it. Okay, and this is much more in the realm of talking in cell biology or developmental biology. But really, these immortalized cell lines, they don't look like their original cells, right? So Keiko2 epithelial cells that you can grow in the lab, they look nothing like the epithelial cells that they were derived from. They have many of the same traits, and they sort of look a little bit like epithelial cells, and they behave somewhat like epithelial cells, but they're nothing like their original state. And so even if you were to harvest polarized cell lines like epithelial cells, or harvest a whole bunch of epithelial cells from mouse intestines, let's say, and I were to culture those in tissue culture, they're going to behave quite differently growing on plastic. So these like plastic or glass tissue culture flasks, they're going to grow very differently in that environment than they're going to grow in the mouse intestine. Okay, so I can try and recapitulate, what they need a little bit and some various things, but recreating what that environment is like in the mouse intestine or in the human intestine, you're not going to really be able to do it. So you try and do the best you can, and you can still learn a lot even with imperfect models. And also, remember that a lot of the surfaces that we've talked about are in terms of epithelial cells, and a lot of the important things that we're going to talk about coming up, these are mucosal surfaces, right? So remember, these are epithelial cell layers that are covered with mucus. So they're covered with this mucin protein. It's a glycoprotein, it's hydrated, there's all kinds of other stuff in there. There's secreted IgA antibodies, there's all sorts of antimicrobial peptides, there's all sorts of stuff in mucus that we cannot really recapitulate that well in the lab. Okay, so there's a lot going on in the host that we can't always capture in the lab, but we can do pretty darn good job. So, summarizing where we went with this last lecture, I want to open up for questions before we move on to our next lecture looking at bacterial secretion. Any questions? How many of you guys have worked with tissue culture? Okay. Yeah, so it's a very powerful tool, but it's not without its limitations. And these are just the ones that I went through are some of the major ones for thinking about pathogenesis, even for studying aspects of human biology, cell biology. There's even more striking limitations. Okay. So, where are we going now? Well, where we're going now, whoops.
Hey, Professor.
Yes.
There's a question on the homework that says, why do tissue cultures allow you to directly study those 3 stages? Can we go over that?
That's for you to think about. I'm not going to give answers to the homework question. Okay. So, bacterial protein secretions. So what do you guys think that means? Bacteria secretes proteins. So what does secretion kind of imply?
Produce.
So you're producing something.
Synthesis. Export.
Export. Yep. So you're producing something, and it's being exported, right? So where in prokaryotes are proteins synthesized?
Cytoplasm.
In the cytoplasm, right? So there's no compartmentalization in terms of replication, transcription, translation. So you're producing proteins in the cytoplasm. So if we were to think back to our picture of a gram-positive or gram-positive or gram-negative cell, let's start with a gram-positive cell. So you have an inner and outer membrane, right? So does the inner membrane have proteins in it? So it has no proteins?
Yeah, it does.
Either. So just the cytoplasmic membrane. Are there proteins in the cytoplasmic membrane?
Yes.
Sure. So you have to be able to transport things. You have to be able to perform electron transport. Two kind of striking examples of things you're going to find in the membrane. So the outer membrane for gram-negative bacteria, are there proteins there? Long story short, yes, there are. So what we're going to talk about today is this is a fundamental challenge in any biological system, but for gram-negative bacteria specifically, where we're going to kind of focus a lot of attention, you're dealing with 2 discrete membranes that both have tons of proteins embedded in them. Okay? And the mechanisms of pathogenesis that we're going to get into after this next exam, and even a little bit of what we're going to talk about on Tuesday with the paper that's now posted, is that essentially how are those proteins getting there? So that's its own incredible structural challenge, right? That you can produce all the proteins you want in the cytoplasm, but how do those periplasmic proteins for gram-negative bacteria, how do they get there? Is it important how they get there? Yeah. And we'll get to some of how that was done. Okay. So, what are we going to cover today? So So the general problem of protein translocation across lipid membranes. So that is one of the fundamental big chunk of stuff we're going to talk about today. And two major ways that exist for gram-positive and gram-negative bacteria to get this done is through the general SEC system, general secretion system, and the TAT system, which is essentially twin arginine translocase system. Then I told you about the gram-negative outer membrane, and that there are a bunch of proteins there. And how they get in that membrane is really a challenging feat, right? And so there's a dedicated system in gram-negative bacteria called the BAM system, or beta barrel assembly machinery system, that I'm going to talk to you about. And then finally, for gram-positive bacteria, their cell wall is decorated with proteins. So it's not just peptidoglycan and some teichoic acid, and there's nothing really going on. That cell wall is littered with proteins. Okay, there's tons of proteins that are being displayed to the outside world by gram-positive bacteria as well. And there's a very unique set of enzymes that are involved in doing this in gram-positive bacteria called sortases. And we'll talk about what those are. And then we're going to get into the first big connection that we're going to see in terms of what pathogens use to be good pathogens are a number of dedicated specialized secretion systems, and I'm going to take you through what those are by the end of the lectures. So, remember that membranes are important protein scaffolds, right? So that is one of their fundamental roles, is they are a scaffold for proteins. And there are peripheral membrane proteins, and that these interact predominantly with the surface of lipid membranes, and they can be non-covalent or covalently bonded to lipids and interact with the membrane. And then there are integral membrane proteins, but these are embedded in the hydrophobic core of the lipid bilayer membrane. Okay? And then that most of these integral membrane proteins, they're directly associated with the membrane, right? So there's kind of no bridge, or that there's not lipids that have been covalently linked to the protein that are letting them sit sort of right next to the membrane. But these things, they are really buried in that membrane. Okay? So the challenge is, one, specificity. What defines what proteins end up here, right? I mean, DNA polymerase is not just sitting in a cytoplasmic membrane, right? It's got plenty of hydrophobic regions of that protein that presumably should be fine hanging out in between the charged head groups of these lipids, right? So what dictates what proteins either get across this membrane or get integrated into it, right? So also, there's the energetic issues and just mechanical issues. Is how does a protein get moved across or into this membrane? How does that get done? And where is the energy derived for this process? Okay? So transport, when we talked about it, you can use proton molar force. You can expend ATP. So these are sort of two real clear ways that we can spend energy to get processes to happen in the cell. So where does the energy to put this protein into this membrane come from? And the problem of size. Like proteins, they range in size. And so would you guess that integral membrane proteins are really small, or are they really big? Got big. Anyone want to say small? You think they should be small? You think they should be humongous and big? Right. So there's no sort of like innate bias that we have to think like, all right, they should be big because I don't have a reason why they should be big, and I certainly don't have a reason why they should be small. So how do these fairly large macromolecules end up embedded in membranes? So the first thing that I'm going to talk to you about is the general secretory system. And the general secretory system is fairly well conserved across domains. Okay, so we do something, our cells do something very similar to integrate proteins into the membrane or translocate proteins across the cytoplasmic membrane. And so when I say this is conserved, at least when we're talking about bacteria, that this is a system that is present both in gram-positive and gram-negative bacteria. So if I was to go look at Bacillus anthracis, which is a gram-positive pathogen, I would find these same components that I'd find in Salmonella, which is a gram-negative pathogen. Okay? So the general requirement, or what's going on, is that there is a membrane-bound secretion machine that is embedded in the membrane and essentially self-embeds itself, which is its own interesting problem, right? So you have a membrane-bound machine that's responsible for putting all of the other proteins in the membrane or moving proteins across the membrane. How did that protein get there, right? So it's chicken and egg kind of problem. It appears this machinery integrates itself into the membrane, which is in its own right. But for proteins that are involved in all sorts of processes that we're going to talk about, what happens is that how do we know that something's going to get translocated? Well, it turns out that the N-terminal region of proteins that are destined for secretion by the general secretory pathway, they have their own signal peptide that's at the N-terminal region of the protein. Okay? So many of you that have taken cell biology, how many of you guys have taken a cell biology course so far? So a good number of you. So those of you that haven't taken a cell biology, ask the folks at your table about this that have taken a cell biology class. But so in eukaryotes, like how are proteins that are translated directed to different organelles, let's say? That there's often peptide sequences on the N or C terminal end of proteins that are signal peptides for various pieces of machinery to essentially shuttle proteins to different organelles, for example. So there's a nuclear localization signal or signals, for example, for proteins, transcription factors that say they translated out the cytoplasm of host cells to get basically directed back into the nucleus. There's a localization signal for that to happen. The same concept is true in prokaryotes in terms of how proteins that are destined to be in the membrane or transported across the membrane, they have signals on their N-terminal sequence. So the same machinery, which is remarkable, does two things. So it makes a decision based on what shuttle protein is going to interact with it about what's going to happen to that protein. So is it going to end up in the membrane, in the cytoplasmic membrane, or is it going to get translocated across the cytoplasmic membrane? So how that decision is made is that there are two different signals, essentially, on the N-terminal of secreted proteins. There's either a signal for Sec-A recognition. So Sec-A, or secretory component A protein, is something that recognizes a particular N-terminal signal sequence on proteins. And essentially, this binds to this signal sequence and directs this complex to interact with the general secretory machinery that you'll find in the cytoplasmic membrane. And what this does then is that this Sec-A bound peptide is destined for translocation across the membrane. Okay? And what happens is that this particular peptide, once interacting with this Sec machinery, essentially roughly around 12 amino acids at a time, is pumped across the cytoplasmic membrane in an unfolded form. Okay, so this mechanism requires that this peptide get unfolded as it's being shuttled across the membrane. Now, how do proteins, and that this process is ATP dependent. Okay, so you're burning ATP every time you're moving 12 amino acids. You're spending about 1 to 2 ATPs. Okay, so beyond just the price of synthesizing a protein, which is expensive. So about every amino acid you're adding is 1 to 2 GTPs, for example. Now you're spending about every 12 amino acids or so, you're spending 1 or 2 ATPs to translocate that protein. So secreted proteins are very expensive. And so proteins that allow for this to happen, to end up with this wonderful mosaic of proteins that are embedded in the membrane, that occurs through a process known as, it's the signal recognition, the particle pathway or SRP pathway. And just like the SEC-A recognition sequence, on the N-terminus of proteins that are destined to be inserted into the membrane, SRP binds to that N-terminal sequence, also directs this to the secretory machinery, and through this interaction, essentially you begin the same secretory machinery with slightly different associated components, begins to secrete and essentially insert this protein into the membrane. So it's essentially unfolding this protein, and sort of, you can think about it like sort of spaghetti. It's just, instead of like extruding this spaghetti up on the outside of the cell or into the periplasm, if you're gram-negative bacteria, this is essentially directing this unfolded protein into the membrane, where it will essentially refold with the assistance of the cytoplasmic membrane. So this is one of the things that makes purifying membrane proteins extraordinarily difficult, because oftentimes you want to overproduce proteins that are soluble in the cytoplasm. It's easy to purify them that way, but integral membrane proteins, they need that membrane to essentially fold properly. Okay, so SRP recognized peptides by the SEC system end up embedded in the cytoplasmic membrane, whereas SEC-A recognized peptides are destined for translocation across the cytoplasmic membrane. Okay, so SEC-A, SRP. Are there any questions so far? Yes?
The translocase that is on the membrane, are those different for SEC-A and SRP?
So the SEC-A recognized system or this SRP recognized system, are they fundamentally different? The core secretory machinery, no. But there are a number of accessory proteins that when SEC-A binds to this, it recruits a number of other accessory proteins to say this needs to get translocated across the membrane, whereas SRP-bound proteins interact with the same core complex and recruit other SEC proteins to essentially engage in this membrane insertion process. Good question. Any other questions? So big nuts and bolts here, SEC-A versus SRP, okay? Now, it would be well and good if every single protein out there could get translocated in an unfolded state and just refold when it's outside the cell or in the periplasm. Okay? That'd be fantastic. But there are a number of proteins that if you unfold them and try and refold them again, it's not gonna happen for that protein. And there are a number of secreted proteins that, for structural reasons, need to remain in their folded form. And it turns out nature has found a way to deal with that too. And in bacteria, the remarkable system is this CAT system. And so this stands for twin arginine transport. And what this does is this machinery, instead of unfolding the protein and translocating it, essentially assembles a molecular pore that allows for a folded protein to be shuttled across the cytoplasmic membrane. So that, in and of itself, I would argue, is a remarkable feat. It's one thing to energetically unfold a protein and move a linear polypeptide across the membrane. That's a whopping problem, right? That's challenging. Now, if you're going to say, I'm going to take this big folded protein structure, essentially this beach ball, and I'm going to move that across the cytoplasmic membrane, that's a different problem, right? So, as such, there is different dedicated machinery, and there are some interesting question marks left in this particular pathway. And understanding the TAP translocase, there's an entire essentially subfield of microbiology that looks at this. But more or less, what I want you to know in this class is that in that N-terminal region, that same N-terminal region where you might find a secretory signal for SACA or SRP, you can also find for proteins that are basically destined for secretion but need to remain folded, they have a different signal peptide that contains essentially a number of things, but the main conserved thing is there are two arginines right next to each other in this particular signal sequence, hence this name, uncreated name, the twin arginine transport system. And what this does is that this twin arginine signal sequence interacts with a membrane-bound complex TATBC. Once TATBC is bound to its protein substrate, it recruits assembly of this TAT-A molecular pore. So it's essentially a number of membrane-bound peptides that essentially come together to form a molecular pore, but then this folded protein is shuttled across the membrane, and in the process is cleaved from its twin arginine tag, and out comes our folded protein, sends that signal peptide. Okay? So big take homes: unfolded, folded. Unfolded, folded. Okay? Those are the really high level, if you take nothing else away from those two descriptions. The sex system is for translocating proteins that are fine being unfolded, or membrane-bound proteins. The TAP translocase, that is for moving folded proteins across the cytoplasm. Okay, so that's getting things across the cytoplasmic membrane. Are there any other questions about that process? Why do you think that might be important? So if I'm a pathogen and I make a toxin, and I want to, and that protein, that toxin is a protein, does it do any good if it's inside me and I can't deliver it to you?
No.
No, right? So, what's one thing that you guys think might get translocated by either the SEC or the TAC pathway? Toxins, right? Okay, so there's a number of things that we're going to see, but this is the main vehicle in how pathogens interact with their host. It's through protein interactions, protein carbohydrate interactions. These secreted proteins secreted by pathogens are the bulwark of the molecular underpinnings of pathogenesis. Okay? So, for gram-negative bacteria, I alluded to this additional challenge, right? That there's an outer membrane. So doing this is tricky, right? Now imagine you have another membrane sitting up here. So you just secreted this protein. Now this protein needs to end up in the outer membrane. Okay, that's a whole nother problem. And so what's interesting and true of nearly all outer membrane proteins for gram-negative bacteria, if we were to categorically look at them, so you'll see what's termed OMPs, because purifying outer membrane proteins from bacteria, or at least analyzing them, is a very straightforward thing to do. It used to be a very old way of typing different serivars and pathogens. So, I isolated 12 different salmonellas from 12 different places in the United States, and I wanted to characterize them molecularly. I could extract outer membrane proteins, display them via SDS page, stain those gels, and look at the outer membrane protein patterns of those and say, okay, well, these 3 look really similar, these 4 look really similar, and the rest of these are like their own thing, amongst many other ways that I could type pathogens that were isolated from different places. So, there's some important properties about these outer membrane proteins, and where it's important to begin is thinking about what we see with integral membrane proteins. So what typically happens when integral membrane proteins are folding in the cytoplasmic membrane is that they will adopt alpha helical or beta sheet domains that essentially allow for stabilization in the membrane. Now, in the outer membrane, those integral membrane proteins, things like molecular porins that allow substrates from the outside environment to diffuse through that outer membrane, these adopt a very unique structure known as the beta barrel. And so nearly every single protein you find in the outer membrane, if I were to crystallize it and find out its 3D structure, will be a beta barrel. Okay? And there's an important reason for that. And when we think about what these outer membrane proteins are doing, one of their important things is that some of them form molecular pores that allow for mass transfer with the outside world. Essentially, all the transport mechanisms that we talked about, say bringing carbohydrates into the cell, those are cytoplasmic membrane processes. And if we think about a gram-negative bacterium, say glucose needs to get across that outer membrane, right? How that happens is through diffusion through big molecular pores. And those big molecular pores are formed with beta barrel proteins. And so specific or solute transport, protein transport across the outer membrane. We'll see examples of cellular adhesion, certain outer membrane proteins, specifically, we'll talk about later, type 5 secretion systems. Those things are hardwired for cellular adhesion, so being able to stick to things. And also there's a large number of proteases that have beta barrel domains and are integrated into the outer membrane. So when pathogens are interacting with proteinaceous surfaces, they're essentially chopping up proteins and also degrading outer membrane proteins that are out in the outer membrane. So the beta barrel proteins, how they get there in the outer membrane is that there is a dedicated set of machinery, the beta barrel assembly machinery, that is embedded in the outer membrane. And so proteins that are destined for this outer membrane, they're delivered via the SEC system in an unfolded state out into the periplasm. And there's a series of these BAM proteins that interact directly with this SEC translocon. This essentially is translocase. And essentially, there are other signal peptides that are on BAM substrates that are cue for this BAM complex to grab onto this unfolded protein and essentially begin shoveling it into the outer membrane, allowing for folding of this very unique beta barrel structure. Okay? So, this is required. This allows embedding in the outer membrane and proteins that are destined for this process and ending up in the outer membrane. These are beta barrels. Okay? So when you see beta barrel structure proteins in gram-negative bacteria, it's a fairly sure bet that those things are going to end up in, or that I would find those things in the outer membrane. Okay? Are there any questions so far?
Are they always open?
Always what?
Are the beta barrels always open? Are they pores?
So when they do form pores, the beta barrel, like if I were going to look top down at a membrane, what beta barrels, I'm trying to think if I have a structural picture of one later, but essentially they're sort of like a big sheet that wraps around in a circle. But the center of that circle of these beta sheets that kind of wrap around, that's not often just a molecular pore by itself. What they do to form pores is that these sort of cylinders have different diameters and will interact with each other with different numbers, and the actual pore is what's in between these cylinders. And so you can have 3 stacked together, and that can be 1, and depending on the diameter of the cylinder and the oligomerization pattern of those cylinders, that dictates the molecular weight or molecular size that can move through that pore. Great question. Good question. Any other questions? Okay. So you'll see an example of a beta barrel protein when we talk about type 5 secretion. So gram-positive protein display. This is a whole other world, right? Because gram-positive don't have an outer membrane, right? So hopefully this picture from a few lectures ago, you see this and you say, gram-positive. Okay. So gram-positive bacteria have a different challenge, right? So instead of this outer membrane, you have a big, giant, thick cell wall. Okay? So it's this big, thick, chunky polysaccharide wall. So you can embed things in the cytoplasmic membrane. You can move things across this cytoplasmic membrane. But how in the world do you get things to interact with this cell wall? Because in general, most proteins that are secreted by bacteria, just remember how expensive those proteins are to make. If you're going to secrete something, it darn well better disproportionately benefit you to secrete that thing, right? So if you just spent 500 ATP to essentially spit something out into the environment, it better either disproportionately benefit you as an individual, or it better disproportionately benefit your collective group that are all genetically similar, right? So those are some important kind of sort of driving forces in thinking about, all right, you're going to secrete something, it's expensive, it better do something good for you. So if you don't want something to secrete and fly away, for gram-positive bacteria, you're going to anchor this thing to the outer membrane. And so how does this get done? Is that you would essentially, you could have a non-covalent interaction with this, but in terms of diffusion, eventually that thing's going to diffuse away. So you want to covalently link stuff to your cell wall, okay? So how is that done? So in contrast to this beta barrel assembly machinery, so what we're contrasting between gram-positive and gram-negative bacteria right now is the outer membrane. If I'm going to embed proteins in that outer membrane, it's a protein scaffold just like the cytoplasmic membrane, and I want to display a protein to the outside world as a gram-negative bacteria. I'm going to stick it in the outer membrane, okay? For gram-positive bacteria, they have the same challenges as a pathogen or just as a bacterium out in the environment, and you want to display a protein to the outside world to interact with something better, to harvest some particular molecule, whatever the reason may be. You want proteins on the outside of you too, and you're going to use, instead of an outer membrane as a protein anchor, a protein scaffold, you're going to use your peptidoglycan layer, your peptidoglycan cell wall to do that. Okay, so how does that happen? It happens through the activity of a family of proteins known as sortases. So what sortases do is proteins get secreted out of the out of the cytoplasm, out into the extracellular space, sort of the space between the cytoplasmic membrane and the peptidoglycan cell wall. And there are a number of these sortase proteins that they're integral membrane proteins that bind to signal sequences of secreted proteins, and they get essentially covalently charged with their protein substrates. And what then can happen in the case here is showing a pillin or pili getting built. But long story short, what ends up happening is that you're able to traffic proteins and covalently link them to the peptidoglycan cell wall through the activity of these sortases. So they are recognizing a secreted protein that comes out of this general sex system. There's a signal sequence that different sortases recognize and essentially shuttle those proteins to the outer membrane, or to the cell wall, and perform a covalent bonding reaction of this particular protein substrate to the cell wall. Okay, so again, kind of in a more direct, close-up picture of what's going on, is that, remember, the N-terminus of a secreted protein, this has a signal, right? And so this is a Sec-A recognized peptide in the cytoplasm, and it's getting secreted by the general sex system. So it's unfolded, and it's getting spit out of the cytoplasm out into this extracellular space, kind of abutting the cell wall. And so as this protein gets secreted, this signal for sortases can be at different points in the protein. Sometimes it's in the N-terminus, a lot of times it's in the C-terminal region of the protein, as shown in this figure. And what the sortase is recognizing is what we would call a sorting signal for recognition by a particular sortase. And so what is marvelous with gram-positive bacteria is there's a general sortase enzyme, and then there are also very specific sortase enzymes that recognize different sorting signals. And so as a gram-positive bacterium, you can say, all right, I have a dedicated sortase system for this family of proteins, a different sortase family for this set of proteins, and so on. So it allows you a lot of functional diversity in terms of what can be done. But they all generally abide by the same core set of mechanisms, that they're recognizing a signal, a sorting signal, a secreted peptide. This is getting transferred to the sortase enzyme and forming a covalent bond with, essentially it's a transpeptidase reaction. You're moving one peptide to another peptide, and that peptide is this enzyme sortase, which then is transferring this same peptide to amino groups in the amino acids that are in peptidoglycan. So it's essentially this series of transpeptidase reactions that now we have our cell wall protein anchored in the cell wall. Okay? So are there any questions about this process? Okay. So you're sure? Absolutely no questions. Yes?
So did you say the sortase could be N or C-terminus, but the others we talked about are usually N?
Yes.
Okay.
And so these SEC and TAT systems, that's an N-terminal signal. Now, if I were to say more categorically what you would see with sortases is this picture, that it's usually somewhere in like the last third of this protein that you're going to find this sorting signal. But there are examples that it's much earlier in the protein. Like all things in biology, we want categorical statements, but there are exceptions to everything, right? Yes?
Does the protein get refolded afterwards?
So does it get refolded after essentially coming out and getting charged onto this? Yeah, so different secreted proteins do this a little differently. So this example here, forming a pilus. So we're going to talk about different types of pili. And gram-positive bacteria, they make these long proteinaceous extensions, just like negative bacteria do. And we'll talk about these in a second. But so they grow these pili by essentially attaching one protein subunit after another using these sortase enzymes. And these are completely folded proteins when this happens. So these things get secreted, charged onto a sortase, and then sort of fold, and then essentially you end up with this transpeptidase reaction attaching this BCP A to BCPB, in this particular example in Bacillus, and you just keep performing this transpeptidase reaction. This is more or less the same chemistry that's going to allow attachment of this big, long polypeptide now, this pilin, onto peptidoglycan. It's the same chemistry. It's just that what these different sortases recognize as substrates is different. But yeah, they can. Often these proteins are folded. Sometimes they're not folded until they're attached into the cell wall. It's all kind of very system specific. But yeah, great question. Other questions? Okay. So I hope that was a little bit of a mental break. So to summarize, general protein secretion for gram-positive bacteria, they use this SEC and TAT system, and essentially proteins that are secreted that we're going to talk a lot about in this class. These things get anchored to the cell wall. So that's how proteins are displayed to the outside world. With gram-positive bacteria, gram-negative bacteria, either the SEC or TAT translocase pathways are used for either unfolded or folded proteins. And that these, if they're destined to be in the outer membrane, they're going to interact with this BAM system and essentially get embedded into the outer membrane. And when this happens, these proteins are nearly always beta-barrel structural proteins in the outer membrane. Okay. So, deep breath. What's next? What's next is this set of things. So we talked about gram-negative sortases and the specialized secretion systems. So when I say specialized, there is a general sortase pathway. There's these specific sortase enzymes, and where we're going to go now is really talking about these type 1 through 6 secretion systems. So, bacteria, they often have very specialized secretion systems, and there are some very important functional reasons for this, because not everything, you don't want everything to always go through the same pathways, and it's often more efficient to secrete things in a very specialized, specific way by having dedicated secretion systems to get proteins outside of the cell. So functions of these specialized secretion systems that we're going to talk about can be secreting proteins to the outside world. So not just I'm going to display a protein, but I'm going to secrete this thing and it's going to float away. Okay? And so things that we'll frequently find being secreted are enzymes. So let's say I'm in a dense carbohydrate matrix and I like to eat glucose, and the majority of this stuff is all, like, if I'm a bacterium around a plant, let's say, I'm surrounded by cellulose, right? And so what's cellulose? It's a polymer of glucose. If I can chew up that cellulose, that's going to be pretty awesome for me because I can just eat everything that gets degraded, right? So what I can do is I can disproportionately benefit myself by letting an enzyme go out into the outside world and break down cellulose into glucose and eat the products of what this enzyme does, right? Other things that we'll see are effector proteins. And among this large family of proteins that we generally call effector proteins are protein toxins, okay? And that is a subject for future lectures. Cellular detoxification. So metabolism is not always perfect. These cells are bacteria are living in often harsh environments. Being able to take bad things that are inside of them and get them out of the cell is really important. Nutrient scavenging and mechanical processes like motility and adhesion. So adhesion is something we're going to talk a lot about in the upcoming lectures. But essentially being able to stick somewhere or move in an environment requires protein machinery that's outside the cell. Okay? And the most famous example for motility is the flagellum, which is it's a specialized secretion system. So, secretion across two membranes, if we're thinking about gram-negative bacteria, this is really hard, right? So it's one thing to just say, I'm going to put something in the cytoplasmic membrane, or I'm going to stick a protein into the outer membrane. But let's say I need to get something from the cytoplasm to the outside world, right? So this is what the big deal with all of these systems is all about, okay? So we're first going to talk about two-step systems because these are easy to partition, break apart, and understand. And then we'll go through these one-step mechanisms, and we'll see these labeled as types of secretion systems. So there are two-step and one-step. This will hopefully become clear in a second. So two-step systems, this type 2, type 5, and the chaperone-usher pathway, what this is getting at is that they rely first on the SEC or the TAT system to get their protein substrates into the periplasm, and then through a number of different mechanisms we're going to talk about, that they get these substrates out into the outside world, either sort of still attached and forming extracellular protein appendages, or that they're secreting things outside of the cell. So the first and I think easiest of these to understand is the type 2 secretion system. And what the type 2 secretion system is that we have to kind of think about these as just being already pre-existing structures. So how these things in and of themselves get built relies entirely on SEC and BAM machinery to put the molecular pore out here and have this intermembrane sort of pump system attached to this pore, right? So our substrate that's going to get shuttled out via this specialized secretion system gets pumped into the periplasm by the SEC or CAT complex, and then is destined to be shuttled out by this dedicated secretion system to pump out this particular protein. Okay? So the benefit here is that all of these mechanisms I'm going to talk about are extraordinarily specific for their own substrates, right? So this is not willy-nilly any protein that these things interact with going outside of the cell. This type 2 secretion system recognizes only its own substrates and only pumps those substrates out of the cell. So these are ATP driven, and I'm going to find some cryo-EM movies that I meant to get up. But essentially, this has been resolved by cryo-EM. And essentially, what this thing is doing is that this is a molecular piston. Essentially, this retracts. These protein substrates get loaded into this pore. And essentially, this piston then expends ATP and pushes these proteins through this pore out into the outside world. And so you can kind of think of this as this ATP-driven mechanical pump that specifically pumps out only its specific protein substrates. Yep.
So it uses sec as that based on the type of single peptide that's on the.
Yep, so the substrate proteins that are gonna get secreted out this pore. So those signals are recognized by SAC TAP complex, but once this thing is folded out in the periplasm, that protein is what's going to specifically interact with this secretion system. Yep, good question. And so there's also type 5 secretion. It's also a two-step process. And what goes on with type 5 secretion is that this protein is secreted, interacts with the BAM complex, gets integrated into the outer membrane, and then forms its own pore. That there is a sort of A subunit of this protein that is the beta barrel pore, and then there is the B subunit of this protein, which auto-transports itself through this pore, and oftentimes auto-cleaves itself, so that this protein that got pushed up through this pore then diffuses out of the cell. And so this, what's remarkable is that this whole process expends no energy. So the energy expended is like putting this thing in the membrane, but this actual mechanical process of this B subunit essentially moving through its own pore, that is a self-assembly process. That is something that is so thermodynamically beneficial for this protein to adopt that structure, that this tail essentially moves through its own pore to get to the outside of the cell. Which, I mean, that is, I think, really, really cool, right? And as we're going to see, a lot of adhesin proteins that are on the surface of gram-negative bacteria, they're type 5 secretion systems. Okay? There's also the chaperone usher system, and what that is akin to the type 2 secretion system, but it is functionally distinct, and that the SEC system is secreting these protein substrates. That this is an example of a pilus that's being formed. That there's an outer membrane molecular pore, and then essentially there is a periplasmic chaperone protein that grabs onto these substrates as they come out of the SEC system, and essentially shuttle these to an outer membrane pore complex that's pumping these subunits out of the cell. Okay? So it's like this, except this type 2 secretion has a mechanical piston, essentially, that's shoving proteins up through this pore, whereas the chaperone usher system, there's a cytoplasmic diffusing protein around the periplasm, interacts with these substrates, brings it to this pore, where this machinery essentially stuffs this new subunit that showed up out of this pore in this growing and assembling extracellular peptide. Okay? So, something that interacts directly with the inner membrane, you have this sort of membrane spanning complex, whereas the chaperone usher pathway is not a membrane spanning complex. Okay? Or basically spanning the entire periplasm. And so this is in deep contrast to what we term one-step mechanisms, one-step systems. These are the type 2, type 3, type 4, and type 6 secretion systems. You see lots and lots of examples of this. But important to all of these is that they're SEC independent. So once these structures are built, they do not require the SEC or TAT systems anymore. So if we think about these as pre-existing systems, that this thing is going to recognize a substrate in the cytoplasm and transport it all the way across both of these membranes in one step. Okay? And the simplest of these are the type 1 secretion systems. And there are a lot of toxins, gram-negative toxins that get secreted using type 1 secretion systems. As you can imagine, you synthesize this thing, this toxic peptide intracellularly, and then you just immediately pump it out of this dedicated pore. Okay? There's the famed type 3 secretion system, and we'll see a lot of examples of this in coming pathogen examples. But what these are is, again, a independent pathway that is structurally distinct from type 1 secretion system, and that it is believed to be ATP dependent in terms of how these proteins get secreted through this assembled membrane, or this double membrane spanning, periplasm spanning complex. The most fun canonical example we have in understanding this is the gram-negative flagellum. So this whole assembly here we can think of, we look at lots of pictures of a flagellum, and related to the flagellum is the injectosome, which is a cellular machine we're going to talk about, that essentially transports proteins from the bacterial cytoplasm all the way across both of its membranes and also across the host membrane to deliver its own bacterial cytoplasmic proteins into the host cytosol. Okay? So this is an absolutely remarkable system, essentially a molecular hypodermic needle that is delivering bacterial proteins into a eukaryote's cytoplasm. Okay? So we're going to see a lot of these. Type 4 secretion. What we often see with type 4 secretion is that it can be sec dependent. More frequently, it is not. And the best example for anyone that's taken microbiology of a type 4 secretion system is the conjugal pilus, the F pilus, that essentially allows for translocation of DNA from one host to another. Okay, so much like the type 3 secretion system, type 4 secretion is another mechanism that will deliver proteins and not just DNA, but it's the ancestral function of all type 4 secretion systems is delivering proteins. So you're translocating proteins from the bacterial cytoplasm to the host cytoplasm. And in one special case, you will deliver DNA from the bacterium into the host, and that's the TI plasmid and Agrobacterium tumefaciens. So essentially delivering bacterial DNA into plants, and that induces a cancerous crown gall growth in plants. And a later lecture, I'll kind of give some insight into that. And the last one that I'm going to talk to you about of one-step mechanisms is the type 6 secretion system. And so importantly for type 3, type 4, and type 6 secretion systems, and I'm sort of announcing these to you in the numerical order in which they were discovered, more or less, is that the type 6 secretion system, what this thing is another mechanism by which you can translocate protein payloads across both the inner and outer membrane and essentially deliver those proteins into the cytosol of a neighboring cell, whether that be a bacterium or a eukaryotic cell. So it's essentially this needle that gets fired out of gram-negative bacteria that have them, and essentially injects proteins into neighboring cells. And the structural origin of this complex is the bacteriophage tail spike, actually. So this is a virus-derived piece of machinery that's been repurposed for protein secretion, which is pretty nuts. So there's wide distribution of these specialized secretion systems and pathogens. We're going to see that they are fundamentally important for pathogenesis. Okay, this is the framework. This is the protein machinery that gets all of the work done that we're going to talk about. Okay, so I'll leave you with some summaries, and thank you for being patient for another couple of minutes.
Yeah.